Yes, that's exactly right. It's decreasing because the more homology you have and the more similar the organisms are to each other, the greater the potential for horizontal gene transfer. And at an extreme, if they're clonally related to one another, they've got a high potential for horizontal gene transfer. But you can't tell that the events have occurred. We have two lefties. I'm watching note-taking. Any other lefties? That's still 1/3. That's pretty good. Half of my family, at least my full sibs, only one, are lefties, because my brother's a lefty. Whatever. Yes?
Hydrosomes and mitosomes. So in the text, you kind of say that they're relatives of mitochondria, but you just kind of leave it at that.
Yeah.
So what are they?
Yeah, so Charlie came along and asked me the same question. And I said, well, let's look it up on Wikipedia. And Wikipedia basically hand waves the same way that I do. One thing he did ask me, though, is the reason for the hydrogens, whatever it is, pronounce it for me.
Hydrosomes?
Is it just hydrosomes, or is it hydrogen, well, whatever. I always want to take out a syllable that probably should be in there. But at any rate, he asked me whether or not the production of hydrogen by these things is the reason that they exist. And I answered that that's probably not the case, that the hydrogen is probably just a byproduct. And then he said that, oh, well, if you think about it in a mitochondrion, a byproduct is hydrogen, right? It's the protons, and then you mix them together with the electrons, and you add them to oxygen, so you end up with water instead of oxygen. I mean, it's the same idea. You've got this hydrogen-based product coming out. And so presumably the hydrosomes are there doing something, such as perhaps making ATP in some capacity, but they're just producing this hydrogen. But yeah, I mean, not that Wikipedia is the highest of authorities, but whoever decided to write it, the article on them, had no better idea of what they do than I indicated in the book. But, sorry.
So we don't know if they're making energy or not?
Yeah, and I also said to Charlie's right there, we came down here today together because we originate more or less in the same location. The problem is that these things are found in organisms that there aren't necessarily high amounts of grant dollars to study. So although people will figure out what's going on and will study them, it's just going to happen at a relatively slow pace. That's my explanation. But it's interesting, we've got these things that seem to be mitochondria, but they don't have a genome. They're structurally similar. Although I don't know the answer to this, we could probably predict that the genes that are found in the nucleus that provide products that are then shipped back to the mitochondria, that we might see overlap in terms of those types of genes in the hydrosome, implying that they, too evolved from a mitochondrial type thing that went through all of the shipment of its genes into the nucleus, but then took it one step farther to the point where it doesn't have any genes at all. Yeah, that whole chapter on symbiosis and endosymbiosis is in a sense a break from the other 4 chapters, where it's just going through, isn't this cool, isn't this cool, isn't this cool? And at the same time, I mean, these are important examples of microbial evolution, particularly the transfer of genes from the endosymbionts to the nucleus, and also just the general types of increasing levels of cooperation that go on between the 2 organisms, such as we saw on a much smaller scale with those phages in that Jim Bull experiment, where we had the 2 phages that were co-infecting that turned into a single phage.
Also from that chapter, you asked, what are the 2 possible fates of products of genes that have migrated from endosymbiont to the nucleus?
Yeah.
Is that just retention and elimination? Or is there something else in there?
Well, okay, yeah, there is something else in there. So the whole idea that they were talking about them as nuclear genes is consistent with them not having been eliminated. So the 2 possible fates are, one, that the gene products can migrate back to the endosymbiont. So when I say migrate, I mean basically they're produced, say, as proteins, and then they simply are transferred back to the mitochondria where they do some kind of job. And the other fate is that they are not being transferred back to the mitochondria, but nonetheless are playing some useful role within the cell that they're a part of. And this goes to the idea where you've got a number of genes that are found in eukaryotic genomes that have a prokaryotic origin, and a fair amount of those genes come from endosymbionts versus the you are what you eat idea. And those genes are not just genes that are being retained solely for the sake of supporting the mitochondria. They get utilized in other ways as well. And this is something that actually in the text was explored in greater detail with regard to, or in the lectures or something, with regard to the chloroplasts, where you've got a certain number of genes in the wild-type cyanobacteria, or the free-living cyanobacteria, and then you've got basically the same amount of gene products, approximately, in the chloroplast, but far fewer genes because they've migrated to the nucleus. But you've also got a bunch of genes that are of chloroplast origin in the nucleus that are producing products that are used in other places within the cell. So the 2 fates are either they're used for the endosymbiont, they go back to the endosymbiont, or they just get used for something else.
Remain in the endosymbionts get used in endosymbiont? Like, how do they get transcribed?
Because there's ribosomes inside of them, and there are tRNAs inside of them, and there are RNA polymerases inside of them. Yeah, I mean, it's very costly in terms of pathways that you have to have in order to display these things. And in fact, I think it is tetracycline, the antibiotic, that is damaging, potentially at least, to mitochondria, because tetracycline acts on, its target is the bacterial ribosome, and we have bacterial ribosomes inside our cells because they're inside of our mitochondria. So yeah.
The double membranes are protected, isn't it?
The 1, 2, 3 membranes. Yeah, yeah. And I mean, with any kind of, if you have you have any kind of drug, you raise the amount of it to sufficient levels, you're going to have toxicity. Tetracycline, one of the toxicities is a consequence, seemingly, of this interference. And this is, I mean, that's an important component of the original argument that the mitochondria, in fact, are not organelles in the way we think of all the other organelles, but instead are these bacteria that are found inside our cells. I mean, they've got two membranes, just like a gram-negative bacterium, and they've got functions that are very similar to what you see cellular respiration in the aerobic or facultative, the anaerobic bacteria. And then they've got this genome, and they've got these bacterial ribosomes that they produce in order to translate their genes. So I mean, that it was an important part of that. And that's why this business with the mitosomes and the hydrosomes, or however one pronounces it, is such a pain and also a big deal, that you can have mitochondrial-like organelles that if you don't need the genes, if they don't need to be producing genes, you can actually get rid of the genome altogether, and you can retain this bacteria-like thing that's living inside of a cell, eukaryotic cell, that in fact doesn't even have a genome anymore. But you can't confirm it based on DNA sequencing, because it doesn't have a genome anymore.
So then how do like other organelles without the endosymbiosis theory have originate like Golgi influence?
Yeah, so there are people that, at least for some of them, like the flagella, have tried to come up with endosymbiosis hypotheses that are less robust, in part because something like a flagella certainly does not have a separate genome of its own. There's no examples that I know of that do. But these other things, the endomembrane system, so in bacteria, when you're producing a protein that gets inserted into the membrane, the plasma membrane, or gets secreted, the ribosome actually docks with the plasma membrane. So you can imagine that, okay, you could have some kind of vesicle-like thing that's a proto-rough endoplasmic reticulum, and maybe you could insert your proteins into that membrane or put them into the lumen, and then maybe you can have a vesicle that simply transports them to the surface. This would be a way of increasing the amount of membrane area that you have without increasing the surface area that you have with the cell. But if you're going to start becoming more sophisticated than that, where you want to send it off to different locations inside the cell, where you want to start modifying these things, then you need to have some place where you're going to do that modification. So you can have this way station of sorts that initially it could have been just a vesicle that gets acted upon in some way while it's going towards the surface or whatever, but then it becomes more sophisticated. So that's how I would explain, in I guess probably simplistic terms, how a Golgi apparatus could come to be. And then there's also the question of where the nucleus come from, and why do we have a nuclear membrane? And that too came from the rough endoplasmic reticulum, because the nuclear membrane is a rough endoplasmic reticulum. You have ribosomes that dock to the outside of that as well, and insert proteins or secrete them into the lumen. I know it's difficult to sit and figure out what your next question is. I'm very aware of how it feels to be, not the one up front. As far as I know, but it hasn't been confirmed yet, the final is going to be in room 609. And 609 is the room where you, it's the room in front of you when you get off of the elevator. So it's just around the corner there. It happens to not have a projector. So I think that's why, presumably somebody's using this room as a classroom, so on that Tuesday. The little red light is flashing.
Moving on to chapter 13. So I don't understand what's keeping us from calling Volvox as the entire colony as one individual, because it
is. It is.
It is?
Yeah. It's just a question of whether we use the word multicellular.
Multicellular individual, that's what.
Yeah. So I honestly can't answer that question. I will speculate that historically, if you looked at it through a microscope, you'd see a lot of homogeneity. And so you would declare to yourself, there's no cellular differentiation here. And as a consequence of that, you would say it's a fairly sophisticated colony rather than a multicellular organism. But I totally agree with you that I mean, just knowing what little I know about it, it certainly seems like a multicellular organism to me. I guess another problem is that there's this gradation in terms of these volvocine, or however you pronounce that, algae, where you in fact go back to something that clearly is colonial. And then somewhere between that stage and what you see, I think you became multicellular. But people describe these things as colonial, so that's why I use the word. You have to go after about an hour? Oh, you don't have to go. Rock and roll.
I'm a cheater tonight. I'm behind in the lectures, so I'm here to partake of everybody's questions.
That's good,
So,
that's good.
there you go.
Well, I mean, you've got a week.
Yeah.
There's nothing quite like listening to lectures on Thanksgiving Day. I remember as an undergrad where I was visiting my grandparents for Thanksgiving, and I realized that it was Thanksgiving Day and it was the first day I was taking off from studying, working, whatever, for the entire semester up to that point. And I was talking to my cousin, who at the time was in law school, and he was like, "Oh, I take every weekend." Like, oh.
Makes you hate lawyers just that little bit more.
I was actually proud of myself. I took this Saturday off from doing work. And I spent the whole day doing a combination of trying to fix a tractor and filling up a large hole with multiple bucket loads of dirt from the same tractor, which was an incredibly painful, too long experience. And I said to my daughter, I didn't work today. And she just looked at me like I was insane. I'm wondering to what degree it's just like focused in on my belly button. I'm sure I'm wandering off. So you're saying that these questions are a public good. That's good.
At least I understood something, right?
Yeah. And if everybody didn't have,
Yeah, if
didn't ask
everybody did,
questions,
it would be bad.
yeah,
We'd all suffer.
it would be all D, or rather it would be P, it would be mutual defection. Yes, mutual defection.
That tragedy will come.
There would be a tragedy of the commons. That's right. That's perfect. Yeah. But you can get away with it because everybody else is cooperating, and therefore, that's great.
I don't think
It's
it's
a
great.
unilateral defection. You don't think it's great?
It's probably not great, no.
No, I mean in terms
It's
of.
probably be much better if I was caught up.
Yeah, okay, so there's potentially long-term costs. So this is an antagonistic pleiotropy.
Yeah, it's gonna damage my fitness next week whenever I have to watch 6 hours of lectures. You do every single quiz, so.
You know, it's kind of like doing a Buffy the Vampire Slayer marathon, only it will be Microbial Evolution. 24 hours.
I guess they set minimum credit hours for grad students for a reason, right? You don't want to
Right.
take more, you don't want to take more than that, like I did this semester.
Right, yeah. Yeah, I guess they set the minimum number low
Mhm.
for a reason.
Yeah.
When I was in grad school, I took courses like Introduction to Differential Equations and all the engineers. And the beauty of that one is I got an A and I was good. And at this point in time, if you ask me what a differential equation is, I honestly could not tell you. I know it has something to do with differentiation. I know it has something to do with manipulating things. And I know it's using all sorts of rules that I'm sure no one cares about anymore because you do all this stuff on a computer. Once upon a time I knew.
Sadly, they still try and make you memorize all these things.
Yeah, yeah, yeah. I mean, it really is at this point in terms of using math. I mean, you just use a program.
What I like is when they make you learn how to do rhyme and sums and all these other things, and they're like, oh, but here's the computer program to do it for you.
Yeah. But I guess you have to have some minimal level of ability in order to simply know that this is the point at which you need to use the computer program. But algebra? You know, solve for this? That's easy, just type in the equation and say solve for this.
Wolfram alpha was like a new thing. Like when I was first getting into calculus, I think, it was like, this is the greatest thing to ever occur.
I'm sure it was a bear actually doing it at that level. Yes.
I got another.
Yes.
So,
I'm trying to sit down without collapsing the chair. Yes.
I'm not sure why, I think just the section is too long, and I just lose interest or something,
Sure.
but the whole thing on PPIs, protein-protein interactions.
Yes.
I just don't get anything out of that section, so could you
Okay, so.
Could you just make that a
yeah,
summary?
No, it's actually not that hard. So the idea is that you've got genes that are less likely to be swapped out and replaced with other genes because they interact with other proteins within the host cell. And those interactions tend not to make any sense except that that's the way it's done inside of that cell. It's kind of like you go to eat dinner at some family that's not yours and they just do things the way they do it. That's the way they do it. And you will fit in during that meal much better if you can do things the way they do things, and much worse if you can't do things the way they do things. And it's completely arbitrary. So when you're coming into this cell as a gene, if you happen to make a product that in order to be functional has to interact with other genes, or in order to allow the other genes to be functional, it needs this thing interacting with them. The more interactions that that gene's product has to be involved in, then the less likely that it's going to be horizontally transferred, or if it is horizontally transferred, the less likely you're going to get a substantial change in what that gene looks like from the horizontally transfer event. Because the fitness won't be high because there's all these protein-protein interactions that have to occur. Now, there are some genes that their products don't have a lot of protein-protein interactions, and these things are relatively easily transferred. So there's at least two different groups there, the ones that do need a lot and the ones that don't need a lot. And then you've got the business of genes that come in that have a higher potential to have, that products have protein-protein interactions, and ones that have a lower potential. So if there are a lot of protein-protein interactions that are required, then those genes that just inherently can somehow, I guess, participate in those interactions, they're going to have a higher likelihood of being able to be acquired by the recipient gene. So this is the complexity hypothesis. The less the potential for an incoming gene has to successfully do what it needs to do, not just in terms of the function of its gene product, but also the interactions of its gene product, the less likely that that gene product is going to be transferred at a given phylogenetic distance between the donor and the host.
So the two categories of genes are just high interaction and low interaction?
Yeah, basically.
Okay.
Yeah. Or interaction and none. And the argument in the paper that's being cited is that the genes that come in tend to have a lower frequency of interactions than the genes that they replace. Lower number of interactions, which makes sense. Again, I mean, you go into a family that's completely different from yours. There's no way that you're going to be able to interact with the same level of sophistication that the individuals that grow up in that family do. And there was some other argument from that paper, and I don't recall what it is. So that's clearly not going. In fact, I think he even say in one of the questions that I'm not going to really burden you with the details of all of this. So, but the idea of that complexity hypothesis is an important thing. That goes beyond the details of that particular paper. It really probably explains the difference between a core gene and a more readily orthologously replaced gene with something that you could recognize as having been replaced. And it probably is the basis of organismal phylogenies, period. The idea that some genes, in fact, are consistently evolving, that aren't independently evolving within a given lineage of organisms. So it's important. That's fine. That's fine.
No one's saying anything, we're just
No,
gonna
no,
go
no,
through
no,
and
no, no,
go through all the questions that I couldn't answer.
Yeah, no, no, no.
Okay.
That's why we're here.
Yeah.
I mean, literally.
Okay. One of them was, what is the consequence of orthologous replacement on gene phylogenies? Is that just mosaic evolution?
Yeah, it's a lack of correspondence between gene phylogenies and organismal phylogenies, aka mosaic evolution. Those note cards, all the terms, all 100 or 95, so I guess it's over 100, isn't it?
It's easier just to keep them in that.
Works for me, it makes good sense.
I started off trying to do note cards, but there was just too many.
And did have to go and get note cards quite a few times.
So when I'm teaching Micro 4000, I average about 50 of those terms per chapter, and I guess over the course of the semester we go through about 20 chapters. So that works out to about 1,000 terms, but I don't require that they know the terms both ways. I mean, I'll give them the definition, and I want them to give me the term. And then I have as bonus questions, flipping them. So it's not as big a burden as it sounds like it is. Now, when I taught Bio 1114 last semester, last spring, I flipped them just like I'm doing it here. And I think I actually had more terms. I had like 150 or 160 terms for them. So they were more or less able to do it. But it's also easier stuff. I mean, not for them necessarily, but it really is easier stuff at that level than what we're doing in here. Anyway. I guess another way of saying that is that I'm trying not to emphasize the terms as much in this class as in that class.
I also wrote all the questions down up there.
There you go. And on the same batch of cards in the same place?
They're in different groupings.
Yeah, okay. But in the same little container? Well, but
I learned by writing things
yes.
down though.
Yeah, I was going to say the same thing.
The more I write it, the more I remember it.
Yeah, and it forces you to pay attention to write something down. And I even found that when I type things, I'm not engaged in the same way, so it's not as useful.
I did a study on that. There's like hardly any linking at all with the typing.
With the typing?
There.
Yeah, there you go. Psychology major. I can definitely attest to that. That's cool. I mean, this is why I write so much, right? I mean, this is why if you learn by writing and you like to learn, then you just end up writing. Band practice. So what's your major?
Animal science,
Oh, okay.
Bachelor of Science in Nutrition.
Oh, okay. Cool.
I work mostly with rumen microbiology.
Neato. I was briefly a nutritional biochemistry major in grad school. Nutritional biochemistry major with an ecology and evolutionary biology minor. And I was like, this is insane. And so I switched my major, but I kept my minor. Which I'm sure very few people in this world have done. And so what's your major?
Same.
Oh.
I'm getting my master's in Genome.
Oh, cool. Neato. I guess I know what you do. I know what you do. And I think I know what you do.
Yeah, microbiology.
I know Jeff Lejeune. He's at OARDC, heads up the animal, he's in the Department of Animal Sciences also, but it's a different campus.
Yeah, I'll be going there to start my research here shortly.
Oh.
But I'm just finishing my classwork here now.
Oh, then you're going to meet him, because I think he heads things up there.
Anybody in Chickens working with Bill Weiss?
No, Joe
Oh, okay.
Hogan. But I know Bill. He's on my committee.
Hello. Do you have questions? We're starting to run dry here.
No, really.
Yeah, so we'll presumably at some point cross paths up there, OERDC. So just, remind me who you are. I've spent a lot of time in that building. And the across from it is the Notobiotic building, where they make things like Notobiotic pigs. And they've got this chain with a hook on it, and I guess they tie the legs together of the sow. They pull it up, and then they aseptically slit into the uterus and pull out the piglets aseptically, and then put them into these aseptic containers, and they've got Notobiotic pigs. And I think other kinds of Notobiotic stuff. They've got all these aseptic Notobiotic cages all over the place. It's an interesting place. And they had a tornado that went through there, 5 years ago, something like that, knocked down a lot of trees.
What is Notobiotic? Sorry.
Oh! It's an animal that has been birthed and raised in such a way that it has no flora unless you add flora to it. And it starts with a G, Gnoso.
Oh,
But I'm pretty sure the G
like
is not.
Gnostic.
Yeah, probably the same root, huh? Lack of something.
So they have like massive problems with digestion and stuff?
They're not healthy. Yeah, you don't want to live that way. But they're not. I don't think it's massive problems with digestion. And that way you can introduce some kind of whatever to them without having the complication of there being other organisms in there. And it's relatively easy to do that with like mice. But you need this crazy facility if you're going to do it with something like a pig or with chickens and stuff like that. Yes, chickens must be relatively easy, because as long as you can disinfect the egg, right? And it must be possible to disinfect an egg.
I don't do poultry yet.
Eggs are porous, so.
Yeah, they are, but I mean, it must be possible to, who knows? Who knows? Because I mean, maybe they actually dissect off the shell and then disinfect, but it must be possible to do it. It must be I don't want to say easier with an egg, but I mean, the fact that you've got this separate thing must something.
Interesting.
Yeah. So you're suffering from a cold? Wasn't I suffering from a cold when I was last year? I think so.
It's been going around campus, I think, in the past couple weeks. It's just about everybody I know or work with has had it.
That's called transmission. It's a very effective cold, apparently.
So has our like increased interconnectability in the world itself, so like with more world travel, has this allowed for an increase, or I should say increase, but an evolution towards more virulence for certain pathogens because of the host availability and presumably susceptibility?
Presumably. And the examples that I grabbed from Paul Ewald's book, where he talks about the evolution of influenza and the evolution of childbirth fever, where he basically, with childbirth fever, the story is that people are gathered together in these hospitals in order to transmit pathogens among one another, but specifically that was transmission from autopsies to the individuals that were then birthing. But with the, even more speculative, with the 1918 influenza pandemic, his hypothesis is that you had, during World War I and the aftermath, these people that were being, if they didn't feel well, were being shipped to these centers, where basically the sicker you are, the more likely you were to be shipped to these places, the hospitals, and therefore the greater the potential for transmission within these hospitals. And maybe that resulted in the evolution of a highly virulent strain of influenza that then was also transmissible between humans and just broke out of the hospitals and spread around the world. But the interconnectedness problem is a huge issue that, and the densities of our cells. I mean, when we were evolving, we evolved in these little clans, and there wasn't all that great a potential for a pathogen to even make contact with the clan. But if it was too nasty a pathogen, it was specifically human pathogen, if it was so nasty that it wiped out the clan before the clan had an opportunity to meet up with another clan, then it would be a dead end, and so it wouldn't persist. So the fact that we've got this ease with which these pathogens are able to be transmitted even when they're making individuals really sick has the potential to result in the evolution of much higher levels of virulence. And probably you're seeing this more than anywhere else in hospitals themselves, where you don't just have an increase in the transmission of existing strains, but actually the evolution of strains that they're more effective and potentially more virulent within the hospitals. Yep.
Simple.
Hope you feel better.
Thanks.
You're welcome. I'm surrounded by poultry at my home. We now have so many eggs. I probably should have brought some. We have so many duck eggs from just 2 laying ducks that we cannot even come close to keeping up with the ducks.
Bring them to the exam.
Bring it to the exam?
I will take as
You'll
many
take
as
some?
you can give me.
Okay.
I love them.
You remind me by email.
Will do.
Did you like pickles?
Sure, I do like pickles.
So remind me about that too. I'd be more than happy to I'm a pickle enabler or proselytizer, I guess. I like to provide people with pickles. I was yeah, however that sounds. I was teaching a class a year ago. I was teaching actually 4 lab sections of Biology 1101, and for the I brought pickles in for the sake of osmosis, right? It's preservative, and I brought pickles in. I couldn't get them at the time, half sour pickles, because they're a product of fermentation. So it's like an osmosis lab and a fermentation lab. So I had 2 excuses to bring in the pickles. And I had at least, this is among 100 students, I had at least 2 students who said they had never in their life before knowingly eaten a pickle.
college students.
College students. Isn't that amazing?
That's good. Well, good on you for
Thank
introducing
you, thank you, thank you.
The American college students?
Yeah.
Well, everybody in my office is from Asia, and so I'll bring in like, I don't know, meals that I think are reasonable. They're like, what's that?
Wow, yeah, okay. But I bet you, I mean, there's all sorts of pickled stuff in Asian cuisine. When I'm not wearing my professor hat, I'm a Szechuan Chinese cook. And I, in fact, just yesterday, because I come down here. Why do I come to Columbus, you ask, for my job? No, it's so I can go to Trader Joe's and pick up more Chinese ingredients. But so I was in the Chinese grocer that's near the Trader Joe's over in Dublin. I remember. And they've just got, you know, jar after jar after jar after jar of pickled stuff. It's not necessarily pickled cucumbers, but there's just a phenomenal amount of pickled stuff in their cuisine. And I get, it must be, I mean, as long as they have access to salt and vinegar. Indian cuisine, I mean, they've got tons of pickled stuff. So I suspect that it probably you almost have to be American to have never had a pickle. But then I figured out a way of getting half sour, halfway decent half sour pickles. Does anybody know or not know what a half sour pickle is? So, yeah, that's right, you do know. That's right, because I brought them in, yeah, that's right. I had to get them in a 5-gallon container. And so I had this 5-gallon, tub of pickles. So there are pickles that are refrigerator pickles, so you add your salt and your vinegar, but they never leave the refrigerator, and you don't have to be as stringent in terms of what the brine is all about in order to keep the normal flora, whatever's contaminating it, from growing, because they're in the refrigerator. There are pickles that you have to cook, so these are essentially canned pickles. And so they, I guess, also you reduce the amount of flora or get rid of it entirely by canning them, and then you have vinegar and salt and whatever. And then there are half sour pickles. And half sour pickles, you put together your brine, and you have this container, and you leave it uncovered, but you put, what's it called? Cheesecloth over the top to keep the flies off. And you let it do its thing. And what happens is the bacteria that are on the cucumber or whatever you're using produce lactic acid. I mean, they grow and they produce lactic acid. And every day you have to go in and you have to take the froth off the top so that you keep it, I guess it's not keep it anaerobic because you actually want it anaerobic, whatever, you take that off. And if you have a buildup of lactic acid in this stuff, the same way that sauerkraut is made. And they call them half sour pickles. They're not as soury as when you use vinegar to make them sour. They're great because they're lactic acid, although they are, I think, somewhat of an acquired taste. Because when you first eat them, you might say to yourself, this is a rotten cucumber. Kimchi. Who's had kimchi? So kimchi is a Korean delicacy, and it's basically the same idea, except they use things like Korean radishes and what else? I guess cabbage.
Buried in the ground.
I think
Maybe they
so.
buried it.
Yeah.
Leaves
Maybe
like
peppers
this.
and stuff like that. Yeah. So for the same Bio 101 class, I show videos of how you make hot pepper sauce by fermenting it. So you take your hot peppers and you mix it with your vinegar and your salt and you put it in a vat and you just let it sit for months. The Tabasco company, I think, lets theirs ferment for 3 years. And so I've got these videos where, a person's talking about it, you just put this aside, and then when you come back, you have to kind of take the mold off the top, and it's cool stuff. Fermentation. And then there are papers on the ecology of the fermentation process, such as of sauerkraut, where you have one type of bacterium replacing another because it's an ecological succession. As one bacterium thrives, it changes the environment in such a way such that another bacterium can now thrive, and so you get these replacements. I don't know if they've studied it evolutionarily rather than ecologically. But yeah, it's neat stuff.
What did we talk about that prokaryotic bacteria?
That,
Swiss cheese?
yeah,
That's
Swiss
Swiss
cheese,
cheese,
yeah.
yeah.
Yeah. Very good. And I show a video on
With
Swiss
pickles.
cheese.
I like Swiss cheese and pickles.
That's right, that's right. I had pickles, and I had Swiss cheese, and I had bread, because sourdough bread from Panera, of course. And I had Indian pickle hot sauce, and I had yogurt, and I had habanero Tabasco sauce, because regular Tabasco sauce simply is not potent enough for me. I might have had a couple of other things, but yeah.
Some sort of meat there.
Oh yeah, pepperoni.
Yeah.
Very good. Yeah. So from Gordon's Food Service, where I buy my 5-gallon tubs of half-sour
Is
pickles.
that where you got that from?
Yeah.
I wonder where the hell you
Yeah.
get a 5-gallon pound.
Yeah, I had to special order it. In fact, I have to special order it sometime early December so I can get it in for my next
Really?
biology class and microbiology class. It's $33 or something like that for 5 gallons, which works out. It's a pretty good price.
You have pickles for the entire semester.
Yeah. And so I'd get these big hunks of Swiss cheese from the same place, making sure I get ones that actually have holes in it, because people will better appreciate fermentation, because there are holes. If there are no holes in it, you're like, yeah, well, it's just cheese, right? And you can make cheese without fermenting it, because you can do it without the organisms. And I also get this pepperoni. I get this big, I don't know, maybe that long hunk of pepperoni, because pepperoni is made by fermentation. You get the meat stuff and you put it inside of whatever its wrapper is. And then you actually, I think, inoculate it with some kind of mold or something like that. It has all typhaid going into it or something like that. And that's how it turns into pepperoni. So we have pepperoni. And then I also show a video where there is, what is it? It's some kind of Asian food where you take like raw chicken and you mix it with this bacteria or something, and then you let it sit out overnight at room temperature.
Yeah, there's a Brazilian cuisine that does that, because there was a kid in our one Spanish class, and we had like a fiesta thing in there, and he brought it in. They do that with fish. It was technically uncooked fish, but it was like.
I think this is actually pork or something like that. Yeah. I mean, it's crazy, but it's fermentation. It's a way to preserve foods. I mean, sour milk.
Makes
yogurt,
sense.
stuff like that.
Somebody made a mistake and figured out.
And was really hungry.
I didn't die.
Yeah.
There's NPR did a review on a book, and I can't remember the name of it, but the whole book was about the fermentation. I think it was done by the same woman who does Wicked Plants, and she has like a whole series of. I think she was the one who wrote it. But anyway, it was really interesting because they went through all that like worldwide. Like yogurt, for example, it's the yogurt that we have in America is one type of like all the hundreds of yogurts that have evolved, like co-evolved around the world. So
yeah, convergent evolution.
Yeah, sort of. I mean, sort of.
Convergent against origin.
Well, it's convergent cultural evolution. And speaking of which, at Trader Joe's they have a chocolate stout in a 16-ounce bottle
It's just not that
for $2. It's not bad.
big.
You don't approve?
Chocolate beers, and they're just not.
Oh well, yeah. If you don't like chocolate stout, but it wasn't. I mean, I brought it home and I put it in the freezer and tried to get it cold enough, but I didn't quite get it to refrigerated temperatures, and it really wasn't all that good before I got it to refrigerated temperatures. But I just put a little